right now. Without you even think about it, trillions of a tiny molecular machines inside your muscles are either resting silently or being called to action. Today we are going to take a deep dive into those machines. By the end of the session, you will be able to close your eyes and see a sarcomere contract step by step. Now, welcome you all. Today's topic is one of the most beautifully and frequently tested chapter in biology, which is mechanism of muscle contraction, especially the sliding filament theory. This chapter is called high yield for a reason. Almost every year, every competitive exam asks at least one to two questions directly from this topic, and those questions separates the top scorer from the average one. So pay full attention. Let me give you the big picture first. Your body has a three type of muscle: skeletal, cardiac, and smooth. Today we focus on skeletal muscle, the voluntary muscle that helps you pick up a pen, walk, or even breathe. Muscle contraction is not a simple squeeze; it is a precise, ordered molecular event. In 1954, Huxley and Andrew Huxley, two independent researchers, proposed the sliding filament theory. This theory tells us that the muscle don't shorten by contracting their filaments. Instead, the acting filament sliding over the thick myosin filament, bridging the Z line closer together. Think of it like pulling two combs towards each other. The teeth integrate interdigitate more deeply, but neither comb teeth get shorter. That is exactly what happens in your sarcomere. Today we will cover the anatomy of the sarcomere, the proteins involved, and the role of calcium, the cross bridge cycle. Now, if the muscle fiber is an army, the sarcomere is an individual soldier. Even every contraction that your body makes is the result of millions of sarcomere contracting simultaneously. The sarcomere is the like this slide tells you, the sarcomere is the functional unit of the skeletal muscle. It runs from one Z line to the next Z line. Understanding the sarcomere anatomy is absolutely essential because is essential because the exam will you ask you which part shorten and which part stay the same and why. Let me walk you through the structure systematically. The Z line, you can find it here to the very far of both the end. Okay, this one and this one, also called Z disc, mark the boundary of each sarcomere. The letter Z comes from the German word Zwischen. Sinbe means disc. The Z line anchors the thin actin filament. This is the it is lined by it anchors the thin actin filament. The A band stands for the anisotropic. This is a darker central band that you see in the electron microscopy. It contains the myosin thick filament in there entirely. Crucially, the A band also has an actin filament overlapping its two ends. The A band does not change its length during the contraction. This is the more important MCQ for this slide. For this slide, the I band stand for isotropic. It is the lighter band and contains only the thin filament, which is actin, no myosin. The band I is bisected by the Z line. During the contraction, the I band shortens. The Z band is found in the center of the A band. It is the region where only the myosin is present. Actin has not reached here during the rest. When the muscle contract, actin slides in and the H zone shrinks or disappears. The M line runs through the center of the Z zone and anchors the myosin filament in place. You can always remember as a band is equals to always constant. Studio you people always confuse a band with I band. Remember, a is always constant. H zone, H is stand for has only myosin at rest. H zone disappear when actin slides in. Now, every great machine has a specific component in the molecular machine of the muscle contraction. The two actors are actin and myosin. One is the drag, the other is the motor. The thin filament is actin. The thin filament is made up of three proteins: F-actin, tropomyosin, and troponin. The F-actin is a polymer of a two helical strand of a G-actin, which is a global actin, wound together. Each G-actin has a binding site for the myosin. These are the sites where the cross bridge form, but only when they are uncovered. Tropomyosin is a long rod-shaped protein that runs through the groove of a F-actin helix at rest. Tropomyosin sits right on the top of the myosin binding site on actin, physically blocking the myosin head from attaching. Think of it as a shield or a lid. Troponin is a complex of a three subunit. Troponin C binds calcium. Troponin I, inhibitory, keeps tropomyosin in a blocking position, and troponin T binds tropomyosin. Troponin is a lock. Without calcium, troponin keeps tropomyosin in its blocking position. The thick filament, which is a myosin, is a large protein with a long tail and a globular head. The head is a business end. It has two critical sites: actin binding site, where the myosin head physically grips the actin filament, and ATP binding site, where ATP attaches and get hydrolyzed to power the stroke. At rest, the myosin head is already loaded. It has undergone ATP hydrolysis, is in the cooked high energy state, holding ADP plus PI. It is ready to fire, but the actin binding site is blocked by the tropomyosin. Always keep in your mind, the entire system is like a loaded gun with a safety on. The bullet, which is a myosin head, is cocked and ready. Tropomyosin is a safety. Calcium is what releases the safety. Troponin has a three. Always remember, troponin has a three subunit. It is always asked in most of the competitive examinations, and always remember this thing. Now, here this slide shows the nerve impulse, excitation, contraction, coupling. Your brain decides to move your finger within a millisecond. A signal travels from your spinal cord, crosses a tiny gap, triggers an electrical wave, and causes a flood of calcium inside your muscle. That entire sequence is what we call excitation, contraction, coupling. The motor neuron, there are for this, it occur in approximately 4 steps. In first step, which is the nerve impulse arrives into first step, the nerve impulse arrives at the neuromuscular junction. Into this, the motor neuron axon terminates at the neuromuscular junction, a specialized synapses between the motor neuron and the muscle fiber, where the action potential travels down the motor neuron. It reaches the axon terminal. Where now the second step is release of acetylcholine. The action potential triggers vesicle in the axon terminals to fuse the membrane and releases the acetylcholine into the synaptic cleft. Acetylcholine diffuses across the cleft and binds to the nicotinic receptor on the muscle fiber sarcolemma. The third step is action potential on the sarcolemma and the T tubules. Acetylcholine binding opens ion channel, causing depolarization of the sarcolemma, the muscle fiber cell. This action potential then spread along the sarcolemma into the T tubules, which is a transfer tubules, deep invagination of a sarcolemma that penetrate the muscle fiber. In fourth step, the T tubules action potential triggers the sarcolemma, sarcoplasmic reticulum, the muscle specialized smooth endoplasmic reticulum to release calcium ion into the sarcoplasm, which is a cytoplasm of the muscle fiber. The calcium concentration in the sarcoplasm releases dramatically. Now, without calcium, the tropomyosin stay on the actin. No cross bridge form and muscle remain relaxed or paralyzed. Calcium is the absolute master switch. Clinical correlation: the drug Curare block the acetylcholine receptor on the muscle. No action potential, no contraction, paralysis. Used historically as a arrow poison, and now medically as a muscle relaxant. Now this slide shows the active site for the actin filament. Imagine a stage where the performer is hidden behind a curtain. Calcium is a stage hand pulling the curtain aside. The moment is the curtain move, the performer actin binding site is revealed and show begins. Calcium binds to the troponin C, which is a subunit of a troponin complex. This is a calcium sensor of the system. When the calcium binds to the troponin C, it causes a conformational change, a physical shape shift in the entire troponin complex. Troponin pulls on the tropomyosin, causes tropomyosin to shift laterally, rolling off the myosin binding site on the actin. The myosin binding site on the actin are now exposed. The safety is off. The myosin heads can now physically reach and attach to actin. This is a start of the cross bridge cycle. Now the sequence in the simple term: calcium released from the sarcoplasmic reticulum and floods in sarcoplasm. Next, calcium binds to the troponin C. Troponin complex changes shape. Tropomyosin rolls aside. Actin binding site exposed and cross bridge formation begins. Relaxation, the reverse process. When the nerve signal stop, calcium is actively pumped back into the sarcoplasmic reticulum by calcium ATPase pump, which requires the ATP. As the calcium level in the sarcoplasm falls, the calcium detaches from the troponin C. Troponin return into its relaxed shape. Tropomyosin slides back to the actin binding site and the muscle relaxed. When there is a contraction, calcium released means passive flow down concentration gradient. When it is relaxed, the calcium pumped back. Active transport requires ATP. Both phases are important. The actin binding sites are now open. The myosin head, which has been patiently waiting in its high energy cock position, now lunges forward. This is a cross bridge cycle and it is the heart of the muscle contraction. Now the cross bridge cycle. This is the engine room of the muscle contraction. 4 steps happening thousands of times per second. Each sarcomere is in perfect synchrony. This is where force is generated. This is a power stroke. The first step is formation. Remember, the myosin head is already in its high energy cock position. It is holding ADP and PI from the previous cycle ATP hydrolysis. The exposed actin binding site allow the myosin head to attach from the cross bridge. The system is now locked and loaded. The second step is power stroke. Once the bound myosin head pivot at its hinge region and pulls the actin filament toward the M line, towards the center of the sarcomere. The swinging motion is called the power stroke. During this phase, ATP and PI are released from the myosin head. The energy stored in the cogged head drives this movement. Think of it as like someone pulling a rope. The myosin head in the hand and actin is the rope. The Z line are a dragger closer together. The sarcomere shortens. Now the third step is cross bridge detachment. After the power stroke, the myosin head is in a low energy position, still attached to the actin. For it to get to let go, a new ATP molecule must bind to the ATP binding site on the myosin. When the ATP binds the myosin head, affinity for the actin drops sharply and the cross bridge detaches. This is why ATP is needed for the detachment, not just for the power stroke itself. Without ATP, the head stays glued to the actin fill permanently. Which is what happens in the rigor mortis. Now the fourth step is ATP hydrolysis and recocking. The new bound ATP is now hydrolyzed. ATP to ADP plus PI. This hydrolysis releases energy that recogs the myosin back to its high energy upright position. The head is now ready to bind to the next actin site further down the filament, and the cycle repeats. The cycle repeats rapidly as long as the calcium present in the sarcomere and the ATP is available. The sarcomere keeps shortening. The Z line keeps approaching each other and the muscle contracts. Students always says ATP provide energy for the power stroke. This is wrong. The power stroke uses energy from the cocked state stored from the hydrolysis in the previous cycle. New ATP is needed for the detachment, not attachment. Analogy: the cross bridge cycle is like rowing a boat. The oar enters water, cross bridge form, pulls back, power stroke, lifts out of water, detachment, and release forward, again recocking. Each stroke moves the boat, which is actin, forward. Now this slide shows the ATP double row, the knee trap. The NEET examiner shows love ATP not because it is the energy currency of the cell, but because in the muscle contraction ATP does two completely different jobs in the same cycle. If you confuse them, you lose marks. Let us make sure what happens. ATP role that in the detachment of the myosin head. After the power stroke, the myosin head is stuck to the after the power stroke, the myosin head is stuck to the actin in the Riger configuration, a low energy, strongly bound state. To release a fresh ATP molecule must bind to the myosin head ATP binding site. This binding reduces the myosin affinity for actin and the cross bridge detach. No ATP, no detachment. Now second ATP second role, recocking the myosin head into energy storage. Once detached, the myosin head hydrolyzes the bound ATP into ADP plus PI. This hydrolysis reaction releases energy which is used to physically recock the myosin head into its high energy upright position. The ADP and the PI remain bound to head like a compressed spring until the next power stroke comes. Rigor mortis, the clinical consequence of no ATP. After that, cellular respiration stops. No more ATP is produced. The calcium is in a sarcoplasm is never pumped back. So the cross bridge form throughout the body. But there is a key. Once formed, the myosin head cannot detach because there is no ATP bind to them. Results: every muscle in the body locks in the contracted state. The body becomes rigid. This is a rigor mortis. It is typically sets in 2 to 6 hours after death and resolve over 40 to 48 to 72 hours as the muscle protein begins to decompose. Now, one liner, rigor mortis is equals to no ATP, means myosin cannot detach, muscle permanently contracted, body rigid. Rigor mortis is not due to the excess calcium causing over contraction, it is due to absence of ATP preventing detachment. Muscle are locked because cross bridge cannot be broken. Now the short this is the short notes. This slide is worth more exam marks than any other slides in this chapter, uh, because many competitive examination has asked about band changes in the almost every alternate years. Memorize this table as if you rank depends on it, because it does. When a sarcomere contracts, means when the Z line move closer together, the actin filaments slides inward over the myosin filament, but the filament themselves do not shrink. Let us go through each structure. I band shortens. The I band contains only actin, as actin slides inward over myosin. The I band gets progressively smaller. In full tetanic contraction, the I band can almost disappear. The Z zone shortens or disappear. The Z zone is the actin free central zone. As actin slides in, actin filaments enter and fill the H zone. The H zone shrinks and may disappear completely at maximum contraction. Sarcomere shortens. The distance between the two Z line decreases as the actin filament are pulled inward. The sarcomere itself is a shorter in the contracted state. A band remains constant. This is a critical MCQ answer. The A band the full represent the full length of the myosin thick filament. Since the myosin does not change its length, it only moves actin. The A band remains the same width, always in the relaxed state, partially contracted or fully contracted state. A band length always constant. Actin and the myosin filament length both constant. The filament themselves do not shorten, stretch or changes. They only slides related to the each other. This is the essence of the sliding filament theory. The golden rule: filament slides, they do not shortens. The sarcomere shortens because the actin slides over the myosin, reduces the distance between the Z lines. Memorize this thing. I band shortens, Z zone shortens, sarcomere shortens, A band constant, actin length constant, myosin length constant. We have now covered the entire mechanism from a nerve signal to the sarcomere shortening. Before we close, let us lock in a power mnemonic and do a rapid fire review of everything we have learned. Calcium clears the path, ATP acts the pull. Let us decode this mnemonic line by line. Calcium clears the path. Calcium binds to the troponin C. Tropomyosin moves. Actin binding site exposed. ATP acts the pull. ATP hydrolyzes myosin recoiled power stroke pulls actin. ATP then detaches myosin. The complete story in 7 steps. Nerve impulse arrives at neuromuscular junction, acetylcholine released, action potential on the sarcomeres travel down the T tubules, sarcoplasmic reticulum, calcium into the sarcoplasm, calcium bind to the troponin C, tropomyosin moves, actin exposed, myosin head loaded with the ADP plus PI forms a cross bridge, power stroke will lead to actin pull toward the M line. ADP plus PI released. New ATP binds, myosin detaches. ATP hydrolyzed, recoiled head, cycle repeat. Key fact summary. A band always constant. I band and H zone shortens during the contraction. Rigor mortis. No ATP, myosin remains attached. Troponin C calcium binding subunit. Tropomyosin blocks actin site at rest. ADP plus PI released during the power stroke. Now the final message is muscle contraction is one of the biology's most elegant mechanism. Every time you lift your arm or take a breath, this entire sequence play out billions of times. The more vivid you can picture it, the better you will perform in every examination.